Hi, we're here at Ishi 29 and we're talking with Greg Hampikian. Greg, why don't you tell me a little bit about your background?
I'm a biologist. I got a PhD in genetics and master's and BS in biology. And about 1999 or so, I had already had my PhD for 10 years. I got working in forensics and enjoyed that. That's a large component of my laboratory, and I also consult now and have some patents. But we also do things like cancer drug discovery and discovering new species of ciliates, these single-celled organisms that a colleague in my lab gets to name when he finds.
That's fun.
Well, it contrasts with, these tragedies, with all this crime stuff, and then of course I do Idaho Innocence Project work, so it's people in prison. And so it's nice to get back to nature and deal with those things too.
Absolutely. Let's talk a little bit about some of the things you've worked on. I know you were involved in a study in 2011, mixture interpretation, and that is still something that is
it's
in the
very
headlines.
dear to me. Yeah, it was this piece in the New York Times this last week. What happened was I got into this because I was having trouble interpreting mixtures. So I came to Ishi and I'd go to the American Academy and I took a course with the director of the Washington lab, all these places. And it seemed to me there was a great variety of ways to do this. And that's nice, to interpret mixtures. You do want I don't want one way, right? I want several different ways. But when you did them different ways, you got different answers. And that troubled me. Again, it's good to have different ways. I don't want to see just one way. I'm sorry to see there's people think that this problem is going to be solved, mixture interpretation, by having one way of doing it. That's not necessarily true. We want lots of ways that all agree, so that if you take a pregnancy test from one company, or you take a pregnancy test from your local pharmacy, they both tell you the same thing. You're pregnant, you're not pregnant. Or you're included in a DNA mixture, or you're not. And what I found out was that's not what was going on. So a writer from New Scientist, or an editor, said that I should talk to Idaho Dror, who was one of the world's experts in cognitive bias at the time, something that I didn't know a whole lot about. And he had already done fingerprints and some other things and done great work. And he finally talked with me and said, well, do you have a DNA example I could try this on? And I was like, I have a real case where I think a mistake is being made. Kerry Robinson in Georgia. He's in prison. He's written me. I've been working on it for several years. And he said, well, why don't we do a study? We'll take that data, bring it to one lab, and have analysts look at it and see if they agree or don't agree. Now, what happens sometimes is that people will talk, like you and me. Humans love to agree, like,
Sure, yes.
We'll find something we agree on and celebrate it. And scientists are the same way. We don't like controversy. Nobody really does. And so we didn't let people talk to one another. We gave it to 17 analysts at one laboratory, all of them proficiency tested, all of them at an accredited lab, all of them trained the same way. Gave them DNA data and said, is this suspect included, excluded, or it's an inconclusive set of data? 3 choices. We got all 3 answers.
Oh.
And, this is someone's life, right? And it's also a victim who certainly does not want to have as a further insult the wrong person in prison. So not only that, but only one of the 17 analysts in our test agreed with the original analyst at the Georgia Bureau of Investigation who implicated Kerry Robinson. And that evidence was the only thing that allowed another man, a co-conspirator who admitted to raping a woman and to shooting people, to say, yeah, Kerry was there with me and he did it too. That co-conspirator is out of prison because he shared information. Kerry, who said, I'm innocent, I have nothing to do with this, is still in prison. So you see how a slight mistake that is going from I'm not so sure to yes, I can make a conclusion can have such a drastic effect. And I was finding that in my own work. So I was happy we were able to publish what I thought I had seen. I had no idea it would turn out. I didn't know it would turn out that weird. 17 analysts, only one of them agrees with the original finding. And so I got excited about this. John Butler, who had been doing these interlaboratory studies for a long time, he and I talked, and I said my concerns about, well, I'd like to see some more complicated questions asked in these mixture studies, and I'd like to see scenarios given, because many times, almost all the time, the lab knows what the scenario is, who the victim is, if they're related, et cetera. So most times I see police reports. So they have context, and Ideal Drawer taught me that context is what drives our bias. And I don't think John, took my advice. I think he's a really brilliant scientist, and he figured out, we'll just make it a little bit trickier. And so he did, in the latest iteration of his mixed studies, Mix 13. And that brings us to this piece in the New York Times last week. I was thrilled. When I saw Mix 13 and saw what he was giving analysts, I was like, this is real. These are the kind of cases I'm talking about. It's difficult. And what Butler and Coble at that time were the two authors I knew, what they both said was, and they'd show this slide, the point of this question, this test, is to see how many labs are going to just say, this is too difficult, I'm not going to answer it, which most of my students would do, and how many would give an answer, and how many of those would be right or wrong. And what they found was that 70% of the labs gave the wrong answer. Didn't say, I can't do this, it's too complicated. Didn't say, we can't tell if you're pregnant or not. Said, yes, definitely, we have an answer, and gave a statistic for it.
Okay.
That's just so telling and so important for everybody to know, for jurors to know, for the accused to know, for prosecutors to know, for defense lawyers to know, for scientists to know. I didn't know it would turn out that way. 70% of labs from a National Institute of Standard Technology study get it wrong in a mixture, falsely include somebody in a controlled study. Now, ours was a real case, and some people would say, well, that's better. But in a real case, you can't know the answer. You weren't there. In a controlled study, you know the answer.
You do, right, exactly.
And I thought this should be a major headline right away to tell people this is dangerous. There are, of course, innocent people who are being convicted this way. When 70% of the 105 American and 3 Canadian labs get it wrong. So I was very disappointed when the paper finally came. In fact, it didn't come out for 5 years. And I bugged John, and I we had a confrontation publicly at a meeting. Well, I said, why aren't you publishing this? If it were train wrecks or airbags or contaminated peanut butter, you would publish. And after that, they did publish, the authors published, but I feel that the paper is so full of disclaimers saying it's only these particular cases, no generalizations can be made, and then this very bizarre warning that because we have an adversarial legal system, these shouldn't be used to imply that that other cases are wrong. And I was like, why do the study if you cannot use the conclusion? In fact, the conclusion begins with a disclaimer in the paper, in John's paper. So I was very disappointed. I think, John's a fine scientist and a wonderful person, better scientist than I am. I think this is a mistake. It may be due to relationships with labs, I don't know, but how could you possibly miss the point of that? So we're going to have a roundtable discussion. And
I'm looking forward to it.
I am too, and John and I have emailed each other, and again, I have great respect for him. This is my one beef with John Butler. But I think it is that important that I wrote a piece for the New York Times about it, because I thought, well, if they're going to hide behind a bunch of disclaimers, somebody's got to say, no, this is real. This is a really good study.
Well, it does bring a lot of questions to light, and I think then the next natural question is, where do you go from there? Where do you think we go from there?
Oh, and I think John and I agree on, we agree on, in the future where to go. I think that labs have an obligation, because these people are still in prison, to go back and correct or check their results in inconclusive cases and in cases where it was close or wherever there's a request by a prisoner. Because you can do that now with probabilistic genotyping. It's a program. There are several available. I used one in my grant. And that same one was one that someone got the problem right with in John Butler's study. There are ways now, without doing any new wet lab work, no test tubes, no DNA. You just take the data and you can check your work. And that's been done. Mark Perlin and the Virginia lab, Mark Perlin owns a company with one of these software programs. They looked back at, I can't remember how many cases, a lot of them. And that's what got me started in this, was I saw that they were able to correct inconclusive results. And in one case, an inclusion, where they said the person's included, where they corrected and said he's excluded.
Okay.
When they published that paper and I saw it, that was when I thought, oh, there may be a solution here as well as a problem.
Which is wonderful if that's what that leads to.
Yes, but I think there is a danger that human beings do not want to look at mistakes that they've made. I'm one of them.
I think we all are.
Yeah. And I think there's going to have to be strong leadership and encouragement. That's not something that I can do. I can bring to light the problem publicly, but it's going to be laboratory directors, politicians, and judges who are going to say, you know what, this has to be done because in the interest of justice, it's not that big a deal. Is every case wrong? Of course not. DNA is a good field, but these mixture cases, in the only, 2 studies where it's been done, mine, where one out of 17 agrees with the original analysis, and John Butler's, where 70% of the accredited crime labs got it wrong, I think we've got a big problem. And I have one case, Kerry Robinson, that was the original case that got me interested in this, that we expect results very soon. And I hope to use those results depending on what they are. But we've got a grant through the Department of Justice, and I want to give all the disclaimers. I don't have the prepared form, but they're not endorsing anything that I'm saying. These are my opinions. But we've used that money for the express purpose of looking at old cases with probabilistic genotyping. True Allele is the program we use by cybergenetics. And we have overturned 5 convictions now.
Wow, it's
and
remarkable.
Yeah, in a year and a half, right? Or in 2. One of them was before the grant. But in any case, these are cases where there was an inconclusive result with DNA, and we're able to look at the data again, get a conclusive result. And by the way, the labs agree with us. The analysts who did the original work agree with us. Or where there was, in one case, in Kerry's case, where I think a mistake was actually made with an inclusion that should have been an exclusion. So we'll see. But the promising part is there's a way to correct this. And these people are not dead. This is not history. These are men and women who are writing people like me from prison saying, it's not my DNA. And are they all telling the truth? No. How do I know this? Because I've tested them. I'll tell you that probably a third of the time when we get down to DNA testing, and I think most of the innocence groups would agree with me, about a third of the time it gives evidence confirming guilt. Now that doesn't mean they're guilty. There could be contamination, et cetera. But it confirms guilt and we drop the case. So yes, prisoners lie. Scientists lie, our mothers and fathers lie.
Right.
Yeah, it's, I think I've seen the DNA. We were all the same. The reason why Idil Dror and other psychologists can publish about human behavior is that the chromosomes and the culture makes us a certain way. We're all susceptible to these things. What forensic science could use are outside eyes, outside of forensic science. Forensic science grew up in law enforcement. Law enforcement, and to use John Butler's word, the adversarial system, is one side of an issue. And when we've gotten experts from outside, National Academy of Sciences 2009 report, they said, get the labs away from the police, right? That is obvious. And the other report was the PCAST report. When Obama was president, it was on his desk. And that was done by outside scientists, not forensic scientists only, principally outside people. And you need outsiders. if you want to check on errors, bring the amazing Randy, bring a magician, bring somebody who can look at data and not want to defend their friends or their own lab, in my case, or something like that. So that's what I'm learning. And I'm saying I'm as guilty as the next person. I must be, because it's human nature. It's not prosecutor or FBI or defense lawyers or prisoners. It's human nature.
Right. It's not an us and them. It's just a natural occurrence that you have to build in checks for.
Yes. Yeah. And every group thinks that they don't need them. So,
That's true.
I go to the Innocence Network meetings, and they don't want me to wear their T-shirts sometimes either, because I say the same things to them. Like, how do you know? Why do you say, the other side is bad? They're the same as us. Everybody goes to work, tries to do an honest day's work. All of us cut corners sometimes. We regret it, hopefully. There are C students in every field, and there are A students in every field. So I just want to see us do the right thing by the victims, the accused, even the convicted, if they're claiming innocence. Let's check. Because I can tell you, I have, 24 friends that were prisoners. Some of them served time on death row, and they're free because DNA proved their innocence. So mistakes are made, even in death penalty cases, which you just think should be done so carefully. But no, we've shown that there are plenty of people who were convicted who have been freed.
I'm sure all of those cases stand out to you just because of their nature, but are there one or two that really...
I mean, it's always the one you have. It's the baby in your arms, right? It's the puppy. So, the one that's burning right now. I mean, I have 3 or 4 that are coming to a conclusion in the probabilistic genotyping grant. But Kerry Robinson's been with me the longest, and he was, I've done everything I can. We looked at the data. I thought he was excluded. I sent it to one of my friends in a crime lab. I said, look, nobody has any money. I'm working for free. Take a look. Tell me what you think. He said he's excluded. That wasn't enough. We couldn't get back to court. Then we went to, Fox News Atlanta did a story on it. They sampled 4 of the reporters, right, at my suggestion. We did their DNA. All of them were included. One of them more included than Kerry, and it was a female intern. And, Kerry's African American. She's a white woman in her 20s. That didn't get anywhere. We had a hearing. It didn't get anywhere. Then we did the test at another crime lab with the 17 analysts, published it in a peer-reviewed journal with Ideal Drawer, one of the leaders in the field. Got nowhere, even though The Economist picked up on it, New Scientist picked up on it. So then I waited, met Mark Perlin, who started Trulio, and I didn't like his work. I didn't understand it. And he's like, read the paper. We almost with academics, you throw a pencil, you never come to blows. But Mark and I just did not hit it off, and he said, read the papers, and he gave me a stack of papers. I went home and read them, and I thought, oh my God, this is an actual solution. It's not going to be perfect. There will be errors. Mark, me, we'll all be gone, and someone will find the errors we made. But and I know that John Butler and others agree, that Mike Coble agrees, I've talked to, that probabilistic genotyping is one way forward. There will still be problems, though. I don't want anybody to be fooled by.
No,
we're never going to solve
being an
it.
Outsider to the industry, but just watching over the last 10 years or even less, everything changes. Mm-hmm. It's really growing. It's building. You learn something new every time you work through something.
Yeah, it's really exciting to come out here and see, especially the number of young people who have posters. Poster sessions are my favorite.
Yes,
You can walk around and talk to people and such great ideas.
Along those lines, what do you tell your students? What advice do you give them? I heard you have an interesting business card game, so I don't know, I just heard that.
End of the night, we compare how many business cards you get, and tell me a little something about each of these people. And I do it too, because we're a social species. It turns out that you don't work in a cave. Very few of us are Isaac Newton or Einstein. Most of us are people who need social interaction. I'm very much a social thinker. I enjoy debate. I think that, I know that I'm not right all the time. In fact, at home, if we have to guess, and in lab too, if you think you remember a lyric to a song even, we always say, well, how sure are you? And then we check, and what I'm seeing is like, I have to be 98% sure to have a 70% chance of being right. These other things where I think I know something, the human mind is very forgetful.
Pretty amazing, right, when you really look at it that way.
I try to put data to just about every part of my life. So. And what it tells me is I'm average. I'm really not, a superior being. My guessing, I'm an aggressive guesser, but not awfully aggressive, and I'm accurate. My accuracy is no better than anybody else in the lab or at home when I'm guessing. Mm-hmm. Yeah.
For some of our viewers, once we put this online, that don't have the technical background, is there an easy way to explain mixture interpretation and when they're reading these articles that are published in the New York Times, what they're trying to say and why it's a problem?
Well, can I promote something I don't make any money off of? I did a TEDx talk where I explain mixtures, I think, pretty well. The way I like to say it is, I did this one time for a prosecutor. He really wasn't understanding what I was saying. I said, all right, look, a mixture of DNA, we're going to take the letters from your name in Scrabble tiles and from my name. We're going to put two people in, and now we have a mixture. And let's see what we can pull out. And I was driving with someone who was very literate, and I said, let's give it 10 minutes and see what we can pull out from these two names. And we had a list that's like Ginger, Marianne, Mr. Howell, everybody from Gilligan's Island was there. Galileo, first and last name. We just couldn't believe it. We have like 50 famous names, and I show that in this TEDx talk. And that's what it is. When you make a mixture, you put in two people, you're going to have thousands, perhaps. And that's very hard for us to wrap our minds around. So, for example, if I say, I have your DNA on this pen that was used in a murder, and you say, what do you mean you have my DNA? I'm like, well, you're not excluded. And you're like, well, I always tell my students, if I say match, you got to say statistic. So you're a match, and you say statistic. So you pretend you said it. Statistic. Great. And I'd say, well, it's 1 in 100,000 people are also included. Now, even something like that, it sounds like it has to be you, but we have overturned convictions that were at that level.
Okay.
And shown they were just coincidence. And when you think about it, if there's, say, 1 in 1,000 people who match it, and there's, 40,000 people in this area, in this town, that means there's 40 people who match it. And that's one way to think about it. Now, if there's a lot of evidence about you, that's a very important thing to say, you're matched. But if it's the only thing, another way to think of it is, well, of the 40 people who match, 39 probably didn't give DNA to this, right? So there's always a couple different ways to see it. One of the big findings of John Butler's and co-authors' study, this Mix 13, is when you look at those stats, and those stats are used, as we're using them, to tell a jury, could this be a coincidence? Okay. One of the big findings was that the stats, even when they're using the same method by name, random match probability or combined probability inclusion, vary over 13 orders of magnitude. And as I say in the New York Times piece, that is roughly the same as the difference between soda change and the U.S. gross domestic product. So what are you going to do with a stat variation like that? It is useful, but you better give the range, and you better show why there's a range. What juries normally hear is one stat. Okay. And when a jury hears one statistic, I think when a layperson, if you tell me something is 1,000 times more likely than something else, I like.
They have a psychological power.
Yeah, I'm like, it's probably
everybody wants
true.
A number, yeah.
Right, but we know that when we work likelihood ratios like that with a prosecution hypothesis, defense hypothesis, even if it's 100 to 1, you just say it's probably inconclusive, certainly at 10 to 1. And that's because we know that there are these coincidences. So when it's a cold case and it's just DNA, those statistics become extremely important. When it's just DNA and maybe a co-conspirator saying something, again. When there's lots of evidence, then it becomes a corroborating piece of evidence, and it's important. But those statistics, when I saw how much they varied, that was another thing. I mean, I'd like to say, if you go to court and the judge says, well, bring me 2 receipts, right? And if one of them says there's $30 of damage to your bumper, and the other one says there are $3 trillion damage, right? Do you just cut it in half, give them $150 trillion? What do those statistics mean if they can be calculated so many ways?
How do you educate a juror? What do you tell if somebody's testifying on a complex analysis? What would you say to them?
I like to bring in a PowerPoint, slideshow when I can, and that's up to the lawyers what to do on both sides. But I just think juries love this. They want to know. They know how important it is. And I can't believe how attentive my jurors are. In states where they can ask questions, they ask great questions. So I think going in with a educational slideshow, and as a teacher, I have the advantage that that's what I do for a living, right? It's like, ask a clown to juggle. I think.
Or to scare me. Yeah.
Or to scare me. I think that some experts are not prepared to do that well, to be honest, from what I've seen. But I think that a lot of them do a good job. But sometimes I've been in court where they just start out with numbers and.
Yeah, sometimes people can just glaze over, and it's just a lot to take in.
They do, and they're trying their best. So anyway, I think education is a big part of it. And I had the opportunity to train some judges in Israel recently this summer. I'm going to Montana to work with judges as well. I think all of the different layers need that. And we keep our labs open to everybody. We've trained the sheriffs, and we've trained the defense attorneys and high school kids. I had preschool kids in once. I've gone to senior centers.
I would like to know what that presentation looks like.
A lot of balloons. You can put a fingerprint on a balloon, like with a Sharpie on your finger, and you blow it up like this. It's unbelievable. I learned that from a third grade teacher. Yeah.
Okay. Well, I do like the way this industry seems very collaborative to me, and people really working together in service of justice. So that's a nice thing.
Well, it's both. Again, it's human nature, and so it's collaborative, but it suffers from all the same biases that cancer research does. Right, or stockbrokers, or we're not special just because we wear lab coats, even if they're white. We look like the good guys in white coats. It's just not true. And I like to tell the people who work in labs, I'm like, I know you're careful, but would you want the sloppiest person in your lab using these techniques on your brother or your sister?
Nobody wants to go to the doctor who got a C.
Yeah, exactly. And so that's why we need systems level corrections. So can I tell you some of the corrections that I'd love to see?
Absolutely.
I'll talk about it tomorrow in my talk. It used to be that there was a rule in a lot of labs, and that is still there, that you do all of the evidence first, right? The pen that killed somebody. And you get it all the way to paper, right? Results, before you bring in a sample of a suspect's DNA into the lab. Now, why is that? As we've gotten more and more sensitive, we can detect now single molecules, right? What's a molecule? It's very small. There could be 100,000 of them here. I wouldn't see them, right? 1 million of them. And so that simple rule, and I'll show a slide tomorrow. It's a picture I took in a bathroom in Idaho. It says, employees must wash their hands when? After using the restroom. Yeah, I don't care that you might have food on your hands when you come in. So that simple rule would avoid a lot of the problems we see in court. And I get paid to be a consultant sometimes, and that's one of the things that I have to correct. Like that would wipe out a lot of the times that I have to show up in court. Just do all evidence first. The other thing is go back and look at these probabilistic genotyping. And the final thing is anything that's subjective, that the analyst is like saying, I think that's blue, because we have things like, they look like pregnancy tests. They detect semen, they detect blood, they detect saliva. It's called serology. anytime you have something that is subjective, take a picture of it and include it in your notes. I can't believe they'll just say weak positive for semen on these little tests that give you a little colored line without I mean, they take I got a parking ticket in Boise, Idaho. They sent me a photo.
Everything's on film, right? Yeah.
I mean, I was thinking I could maybe dispute it, but there's a picture. And I go to court sometimes and they say, oh yeah, there was semen in it. I'm like, well, where's the photo? Oh, we didn't take a photo. I'm like, well, you can look under, you have microscopes? Yeah. We saw a sperm cell. I'm like, you saw a sperm cell? Where's the photo? And that's just
So sometimes a simple
sloppy.
fix, really, just
That's
a procedure.
I would just say, I mean, I think those are the things. Do all the evidence before you do anybody's reference sample, which the best labs still do it that way. I was sad to see the FBI not doing it that way recently. And go back and look at your old mixture cases with probabilistic genotyping, and then take pictures of everything. There's plenty of other small recommendations. I think you should, provide stats several different ways so you can show people there's a variety of numbers here. And the recommendations are now coming down from SWGDAM. But we should, anytime there's disagreement in the lab about a result, that should be in the report. Everything should be in the report. Oh, one more. Can I give you one more?
Sure, one more.
We have this technique where we separate sperm cell DNA because sperm cells are very tough. So if you have a vaginal sample, let's say from a sexual assault, or an oral sample or an anal sample, there's going to be epithelial cells from the victim and the perpetrator. These are little cells, look kind of cornflaky, and they break very easily and give up their DNA. So we have a technique called differential extraction that all the labs have been using for decades. And basically, they break open the epithelial cells, suck off the DNA, call it epithelial, then they spin down and wash what's left, and you get the sperm cells if they're there, and you break them open and you get DNA from them.
Okay.
You refer to those two tubes, the ones of the epithelial as the epithelial fraction, and the other one as the sperm fraction. I think calling it sperm, unless you've seen a sperm cell, or at least have tests for semen, is disingenuous at most. I mean, somebody named this. You should just call it the first fraction and second fraction. If in the second fraction you see a sperm cell, by all means, call it the sperm fraction. But I've seen, again, cases where they'll just refer to something as sperm throughout.
Right, which might create bias just indirectly when someone hears that.
Yeah, and I know labs that are actually calling things positive for semen because they've gone through this Six Sigma training. Have you heard about this?
Oh, absolutely, yes.
Right, yeah, because you work for a business. Thank goodness the university hasn't done it. They probably would have eliminated me as redundant. But, so this is an efficiency-driven system, and what I heard from good friends in a lab, people who trained in the same lab that I trained in, we were having a drink I think it was at this meeting a few years ago, and they said, well, everything's positive now for sperm. And I was like, what do you mean is it positive, always positive for semen? And they said, well, you look at it and you're like, and I'm like, what are you talking about? And it's because they went through Six Sigma, and their efficiency experts said, well, if you have child's clothing from a sexual assault, let's say, and you think there might be semen on it from an attacker, it tests for the semen, and if there's semen, send it on to DNA, which is kind of expensive and time-consuming. So screen it for semen. So these honest, hardworking analysts, thinking that they were doing the right thing, were trying their darndest to hold those cards up, to make it look like
to go to that next level of DNA regardless.
And I said, well, why are you doing that? And they said, because once it goes to DNA, you can tell whose DNA is on that child's underwear. And if there's no DNA, it'll tell you there's no DNA, and the person will be released. I said, you're not even thinking that most of these cases that I handle are family members, right? Or people who care for the children. They have access to the children. It's not a stranger. And there may be really good reason for their DNA to be on clothing, if they're folding it, if the kid's on their lap, if they sleep with them, whatever. Not a very good reason for there to be their semen on this clothing. So they were sending it on saying semen positive, and in court, that evidence, that DNA profile, is referred to as the semen from the child's underwear through the court proceedings.
Right, and then if you are implicating a family member,
it's over. Mm-hmm. The difference between holding the card up to see that it's that you can pass it on for DNA analysis. Now, I told you, these are people who are better scientists than I am.
Right, right. And they do think, it sounds like they were trying to do the right thing to get to the next level, but in the process.
Yeah. They're cutting corners,
Yeah.
and it just shows you human nature is like, and I mean, these are really smart people. I was like, oh my God, because they don't see what I see, which is people writing to you saying, I was not there, I didn't touch him, it's not my semen, I didn't do anything, blah, blah. And I have to think through these problems as to, well, what could have gone wrong, and then test it to the best of our ability.
Which brings up a great point. By bringing everybody together, like, what have your experiences been at this meeting? I know you've been coming for, when did you start?
Oh man, I don't know. It's got to be 12 years.
Okay.
I think it's great. I think again, I think we were heading in the right direction for a while, and I think we've retreated a bit. And the right direction is you need outsiders here. You don't want everybody to be law enforcement. More and more, and not just people selling to law enforcement, So we need to have academics involved, and the only way you're going to get academics involved is there have to be grants for people like me. And I've been fortunate. I got some money from Department of Justice and from some private foundations and all. But for the most part, it's like biologists are not going to really touch this subject because it's very specialized. The review panels, the journals, they're all filled with people from law enforcement, or they trained in law enforcement or whatever. And I think that no matter what, that's going to make you clubby. I would say the same thing if they were all defense attorneys, because, like I said, I go to those meetings and they are used to me raising heck with them too. And in fact, I'll say that a lot of the people in the innocence movement at first didn't like probabilistic genotyping because some of them came from a defense background, I think. Okay. They have some legitimate complaints about it, but I think a lot of it's just reaction to, oh my God, that's the newest thing from the prosecutors, and it's only meant to inflate match statistics, and there's that. I'm like, well, no, it's actually very solid science. So, I think we need to have outsiders at any conference and in any field where you expect to get the good debates, like hopefully John and I are going to have tomorrow at the roundtable, or discussion, I should say. And that's what we would benefit from. More of the National Academy of Science type studies. And I think under this, in this particular time with the folks who are in charge of the government, I think that there's less of an emphasis on outside science, more of an emphasis on law enforcement. And there's reasons for both. But I'd like to see more science. I mean, my side is science, so.
Any favorite memories if we could talk about the show a little bit, I'd love to hear about that.
Oh yeah, well I have gotten to sit here once before.
That's right.
Had a great time, and I watched everybody that year, and I probably did for a couple of years, and then I like everybody, you get busy, and you're not involved, like, oh, my picture's not there.
I'm glad we can bring you back in now this time.
Well, you'll have one more viewer.
Yeah, great.
My mother's not around anymore, it'd be two. But I just love hanging out with the scientists and talking to people. The young people are great because they're just full of new ideas, and they're here to throw people like me under the bus or out the window or whatever they're supposed to, and they give you good challenges, and it's fun. And of course, then there's the folks who are even older than me who've been around so long and have such incredible stories about what's happened. So it's meeting people for me that's the fun part of this meeting. And I'm supposed to be in a training session now. I've walked out to do this, so I get training as well. Yeah. So my students are taking notes. So hopefully.
Perfect. Are you going to go to the reception tonight?
Oh yes, yeah, I hope so. Yeah, I have to see. I'm here with two others, so I have to make. Okay. We have one car.
Okay.
Yeah. But we've loved it. It's always in a nice place and stuff. And you guys do such a great job. And we've received help when we couldn't, when we needed help. So in fact, this year too, we got a little help. So I just really appreciate the meetings. I think it's great. And to me, it really is the premier place for forensic DNA. The DNA side, you guys have the meeting. The American Academy, also very good, but it's so many different things that not all the biologists go. But this one, you get cutting edge forensic information. So I love it, DNA forensic information.
I think you would do hear that a lot with the focus, which I think is nice for people, yeah.
Yeah.
Yeah, and I'm glad your students can attend too, that's great.
Yeah.
Some students, yes.
Yeah. I mean, I have a lab person and then a person who's studying. Yeah, so it's great. I'm thrilled to be able to bring people to the meeting. It's just wonderful.
Thank you so much for sitting down.
Thanks.
Is there anything we didn't cover that you want to make sure we do include?
Gosh, you know, gosh, I don't know what else. I mean, the other thing is that you guys are great because you do more than forensics also. So it really comes from that strong background in molecular genetics. And actually, John Butler was just at Boise State last week. I heard one of his 2 talks, and he said something really interesting. He said that he thought the field is moving ahead so quickly. It's because of the molecular biology industry.
Interesting.
Yeah, and he really credited industry, and I hadn't thought of it that way, but it's definitely true. The industry is what's pushing us because there's people making money, make jobs. You know, there's a lot of American jobs here in this field. So yeah, so now I have a new respect for all of you.
Well, that's good. Well, we appreciate you coming back in.
Great. Oh, and also I should say one other thing. I also invite all taxpayers to come visit the lab because for some reason I've been given this glorious opportunity to be a thinker and a tinkerer in science. And I never forget that is just the greatest honor and the greatest opportunity. So yeah, we have lots and lots of visitors come through lab. And even if you're not a taxpayer, you know.
So even if I'm behind, I can come in.
Even if you're behind or you need help from the government, come anyway. You're all welcome.
I think that's actually a wonderful thing to do. I think people are hungry for more information. There's a reason why shows like CSI, whether they're accurate or not, that would be a whole another discussion. But that's why it's popular. People love the field. They're very interested by it.
And those are my heroes. You know, I mean, I've seen Barry Scheck from the Innocence Project give talks to very small groups one that I invited him to, I was so embarrassed. And then to giant places. But these folks Henry Lee, who would speak everywhere, and who I just saw him in United Arab Emirates when we were both there.
We'll have to talk about that. I'm going to Dubai.
Oh yeah, great. And it was wonderful. And and John Butler coming out and talking to our students twice just last week. So I try to pick up from them, even when I'm tired and scattered by all the different things going on. I think, well, no, you have an opportunity and a responsibility. Like all of these people I mentioned have been given access to, so much. I mean, really, to have a lab at a university is a very expensive thing. And there's all these wonderful people who have no idea what I'm doing, right, who pay taxes. So I'm very grateful. Yeah.
Thank you so much, Greg. This is great.